Okay, let's begin. Welcome back, everyone. Some of you are present physically, others are online, and then others will have to watch the video, but I'm going to get started. Okay, we'll begin as always in the name of the Father, the Son, and the Holy Spirit. O Lord, send forth your Spirit, and we shall be recreated, and you shall renew the face of the earth. Amen. So, welcome back. This is the course the bioethical issues at the beginning of human life, our second lecture within the program of the Master of Science in Bioethics. And today's lecture is on fertilization and early embryonic development. We're setting up the biological background for the ethical analysis. Okay. So a very brief review of what we covered last time. First of all, if there are any questions or comments from anyone, feel free to jump in, okay? So last time I was setting up the whole background of reproduction as it happens in nature, because even further back than that, if we are walking around and we see a thing, something, and the thing is metabolizing and has capacity for reproduction, then we're looking at a living thing, organically speaking, okay? And scientifically, we call those species, of which we have discovered on Earth about 2 million of them, but it's estimated to be anywhere between 10 to 20 million species that are existing on Earth today. So the vast majority of species have not been discovered yet. We believe that they are most likely microscopic, and many of them in the deep ocean floor, so it will take a while. However, back to the living organisms, a characteristic of all living organisms is the capacity of reproducing, to generate a next generation, individuals, progeny, that look like the parents and that belong to the same species, with the same capacity for reproduction also. And what we see there is the perpetuation of the species in time, okay, on this Earth, which is benevolent because it has mostly liquid water and light, which are the precursors for the possibility of organic life. All right, so focusing on reproduction, the whole issue here is to establish how reproduction occurs, specifically sexual reproduction in the human, so as to be able to determine when human life begins. In other words, when a new individual begins. And again, we allow nature to tell us that, because that is a fact, that is a biological, organic fact that is independent of whether we believe it or not, understand it or not, whether we like it or not. That's how it's happened. That's how we were generated. And so it gives us objective proof, fact of when an individual human life begins, okay? You can understand that how we determine or when we determine human life to begin has immense impact on the bioethical issues with regards to the beginning of human life. Let me dim the lights also, so hopefully there's enough outside light there. See? No. You have enough light for notes? Yes? Oh, okay. Yeah. See the screen better, right? Okay, so very quickly, just a very brief review of last time. There are two modes of reproduction in nature, two modes of reproduction, right? Asexual and sexual reproduction. And then I have given you examples of asexual reproduction. There are many different kinds of asexual reproduction. I'm just going to move through the slides quickly here. Many different examples. But we are interested in sexual reproduction, okay? So before we go into sexual reproduction, which is actually today's lecture, together with early embryonic development, I also talked a little bit about DNA genetics, because it turns out that this DNA is the universal molecule of inheritance, the universal molecule of inheritance. What is it meant by that? It means that this DNA molecule, which is a huge molecule, has millions and millions of atoms in it, is able to contain the genetic information of every species on Earth, of each species on Earth, so as to pass it on to the next generation. Okay, if you consider that a little philosophically, it is amazing. It is spectacular. We call it in science, it is elegant, that this one molecule, that is an extremely complex molecule, is able to hold the information, what we call the genes, of every single species, so that the progeny, the new individuals of those parents, come out to be of the same species. All right? In other words, there is fidelity in the DNA molecule from one generation to the next. And when we observe these 2 million species that we have discovered, anywhere from bacteria all the way to the largest animals on Earth, which are the whales, we see that in every single instance, it is the DNA that is passing on that inheritance. All right? And that's why we call it the universal molecule of inheritance. It is extremely complex, like I said, millions and millions of individual atoms that compose it, but it has a very regular and ordered structure. It is super ordered, right? And it is in that order that the information is contained. What we call the double helix looks like a twisted ladder, as you can see here. And then the rungs of the ladder, the steps of the ladder, are the ones that contain the information. We call them base pairs, and there are only 4 bases, if you remember from last time, all right? There are only 4 bases, and their bases are also called nucleotides: adenine, thymine, guanine, and cytosine. They're always represented by the first letter, A, T, C, G, and A always pairs with T, and C always pairs with G, so that we have the base pairing. And it is in the particular sequence, of those bases, that the information is stored, all right? So the information is stored in the particular sequence of the bases, sequentially, just like letters on a word, and words on a paragraph, and paragraphs on a page, etc., etc. So it's a chemical information that is all there, chemically, mechanically, so that there is no magic here, and it is passed on from generation to generation. How? Through the process of reproduction. So I think that's sufficient for the review, unless there are any questions or comments. All right, then let's move on to today's lecture. A lot of this, again, I'm trying to bring everyone up to speed on the same page. For most of you, this is very familiar material. Today we're going to cover sexual reproduction and early embryonic development, and just touch on the ontological status of the human embryo, if we have time. Otherwise, we'll pick up on this third point at the beginning of next Saturday, okay, next lecture. In other words, the ontological, the meaning of the human embryo, right? We can determine, is the human embryo indeed a human being, a human individual, a living human being, and if such, then should he or she be protected by law, right? Like the rest of us. So, with regards to sexual reproduction, unlike asexual reproduction, when we only have one individual splitting into 2 essentially, in sexual reproduction, we have kind of the opposite. We have 2 cells fusing into 1, all right? So a little bit is like the opposite of asexual reproduction, in the sense that sexual reproduction involves the fusion of the coming together of 2 cells, but they're very special cells. Those cells are called gametes. And the big picture, if you will, and the bottom line of the process of fertilization is to restore the diploid number, of the new individual. Because remember, the vast majority of cells in our body are diploid, right? In other words, we have chromosome pairs. How many pairs? 23 pairs. Therefore, a grand total of 46 chromosomes in every single cell of our body, except the gametes, which have unpaired cells have unpaired chromosomes, meaning only 23 chromosomes each. And some very specialized cells that have actually lost their nuclei, like the red blood cells. Excuse me. Okay, so these gametes are the sex cells. And of course, we know for sexual reproduction, there are sperm in the male and ova in the female. One ovum, several ova. So this is the added value of the course, that you're learning a little Latin and Greek as we go along. Okay. So these ova and a sperm, these gametes, these sex cells, are produced or generated by a very special process that is called meiosis. Meiosis. Because in this process, what needs to happen is that these sex cells need to lose the pairing of the chromosomes, need to lose the pairing, all right? So that they only have a single set, in other words, the 23 chromosomes, but there are no pairs. And therefore, meiosis is going to be more or less like a double mitosis, a double mitosis. If you remember, in mitosis, from one cell, one mother cell, we end up with two daughter cells. If we repeat the process again, each one of those two daughter cells splits again, we end up with 4 granddaughter cells, if you will. So in meiosis, that's the process. From every mother cell, we end up with 4 granddaughter cells, okay? After 2 splits, after 2 generations. So let's review mitosis for a moment, especially at the chromosome level, at the DNA level, because that's the core of it. That's what the action is. Okay, so this is just straight up mitosis, a simple split of the cell, as every cell in our body is splitting when it's not metabolizing. The cell goes into what we call the reproduction mode. We know that when the cell is in interphase, in other words, not reproducing, when the cell is in interphase, the cell is functioning normally. We call metabolism. The cell is metabolizing, right? So for example, pancreatic cells are doing their pancreatic work, which is mostly producing insulin, and the neuronal cells are doing their work, which is thinking and coordinating all the functions of the rest of the organs in the body, and so forth. Each cell is metabolizing, is doing their function in the body. That's during interphase. And the DNA structure, the shape of the DNA during that time is a single long spaghetti-like thread that we call chromatin, all right, chromatin. And it's extremely long. If we were to pull out one single chromatin from a cell in our body, from any cell, and we were to stretch it out down to the double helix. In other words, not unwinding the double helix, but unwinding the supercoiling, right? And just stretch it out like a twisted ladder only. How long would it be? And if you remember, I mentioned it would be about 2 meters or 6 feet, right? 2 yards, 2 of these. 2 of these. So that's extremely long. How does that fit inside the nucleus of every cell of our body, which is super microscopic? Is because of this compaction that happens, this supercoiling, coiling upon itself. It's like taking a rubber band and twisting it until it coils upon itself into a little tiny wad, okay? That process is called supercoiling, and also compaction, DNA compaction. All right, now that cell periodically has to split because of the volume to surface ratio issue that we talked about, and it goes into reproductive mode. The reproductive mode is what we call mitosis, right? The first thing that happens there, welcome up there, there's a handout there at the entrance. First thing that happens is that the chromatin splits into segments, into distinct segments that are going to become the chromosomes, okay? So the chromatin splits into segments that become the chromosomes, and Then they compact even further to the last moment of compaction, which is the final structure of the chromosome, which is the most compacted DNA structure that we have. And it will be the distinct number for each species. In our case, 23 pairs, right? Okay, so this is an example here of a cell that is undergoing simple mitosis. And there are 4 stages to that mitosis: prophase, metaphase, anaphase, and telophase. They're not necessarily responsible for the phases themselves, but just to know what happens functionally, here is a cell has how many chromosomes total? 4 chromosomes, right? And they happen to be pairs because this long one is about the same size as this other one, and A and B are also supposed to be the same size, all right? So we have 2 pairs of chromosomes, total of 4. Let's move forward and do that. So we'll number them pair 1 and pair 2. So we can talk about individual chromosome 1A and chromosome 1B and chromosome 2A and 2B. Okay, this is just so you follow the arithmetic and don't get lost in the process. You notice that there's a detail here that these chromosomes, any one of these 4 chromosomes, is already looking like an X shape, right? That means that duplication of that chromosome has already happened. Duplication, which we call scientifically replication, right? Because remember, in science, we cannot call things just normally. We have to give it an exotic name. So it's called replication in science, which means that temporarily, we can really say that this cell here in prophase temporarily has a 4n number. 4n temporarily because each individual chromosome has duplicated, but they have not, those duplicates have not separated yet. They have not separated, they are held together by the little white dot, which is called a centromere. A little too much detail, but that's called a centromere. It's holding those duplicates together, all right? So that we speak of each one of those duplicates as individual chromatids, Again, an excess of nomenclature here. But basically this temporarily is a 4n stage, because if we were to count the individual chromatids, we would come up with 4 times 2, which will be 8, you know, 1 and 2 in 1a, 3 and 4, 5 and 6, and 7 and 8, all right? It's a temporary process. What happens then in the next stage is that these chromosome pairs will line up at the equator of the cell, the center of the cell, at the equator of the cell, and they will line up singly, in other words, just the duplicates, because they're still being held by the centromere, but they line up singly, all right? You'll see what I mean by that in more detail when we go into meiosis. Here, the chromosome duplicates line up singly. In other words, here is 1A, here is 1B, here is 2A, and here is 2B, all right? Then the next thing that happens is that the centromere gives way and the individual chromatids migrate to opposite poles of the cell, opposite poles. You see them here in the migration, and then in the final phase, the nuclear membrane is restored, is resynthesized on both new nuclei. And finally, the cell pinches in the middle, the cytoplasm pinches in the middle, which is called cytokinesis, right? The dynamic of the cytoplasm pinches in the middle until we get the 2 daughter cells. And the 2 daughter cells are essentially identical to the original mother cell, meaning that the DNA is a replica of the original DNA. And the cell will also be diploid. In other words, it will have chromosome pairs, all right? Now, a little bit in detail here, because I mentioned that from the interphase to the prophase, the chromosomes had duplicated, had replicated. That happens in interphase, and it's shown here in this diagram. You can see in this cell, which is diploid, because here's 1A and 1B, and here is 2A and 2B. So there are a total of 4 chromosomes or 2 pairs of chromosomes, all right? These 2 pairs, each one of those 4 chromosomes is going to replicate so that before, here's the individual chromosome, and now after the replication, here are the chromatids, the sister chromatids. When it's looking like an X, what we have is 2 sister chromatids that are held more or less in the middle by this centromere region, okay? So that process, as I mentioned, is called DNA replication, and here are the sister chromatids. There are 2 sister chromatids with 2 arms each, so a lot of 2s. Anyway, this happens during the synthesis phase of the cell cycle. This is called the cell cycle. And the cell cycle essentially has 4 parts. The first one is called G1 for gap 1, gap in between reproductions, okay? So gap 1, and then the S phase is the phase for synthesis, which is yet another name for replication. And then there's gap 2 after synthesis, where the cell checks to make sure that the replication has occurred faithfully and so forth. And now the cell is ready to undergo reproduction itself, the reproductive mode, which is the mitosis process, which also has 4 stages and so forth. The end result is the 2 daughter cells, which will then go into the cycle again periodically, unless the cell goes into arrest. In other words, it will not reproduce anymore, like a number of cells in our body actually do that, typically the neuronal cells, which will last for years and will not reproduce anymore. And that's called G0, GO or G0. The cell has gone into arrest and just metabolizing until it will die of old age. And that death process, it's a natural death process of the cell. It's called apoptosis, just for completion, not to worry about it. Now, I'm interested in the numbers here, because again, going to the bottom line for a moment, in mitosis, the diploid number is maintained. In other words, the chromosome pairs. However, in meiosis, there is what is known as a chromatic reduction. Chromatic reduction means that the diploid number is lost. And the granddaughter cells, okay, the end result of meiosis, after 2 splits, is that the cells, the granddaughter cells are haploid. Haploid meaning they are unpaired. It's just the single set of chromosomes from 1 to 23 without any backup. And that already tells you that the gametes, these sex cells, these haploid cells, are delicate, genetically speaking, because they don't have a backup. If there's any damage to that DNA and that cell gets to fertilize or be fertilized, then that damage on average will be passed on, which would generate some kind of abnormality, okay? And so the functional issue with this, with the haploid cells, with the gametes, is that they are genetically delicate. And that's why they're basically hidden in the body, in the case of the female, or in the case of the male, they are in super abundant excess, essentially like 100 million sperm for every ejaculate. Okay, give you a little bit of the punchline, but you see here then in mitosis is 1 split, but because duplication, replication has occurred prior, then the daughter cells end up with a full complement of chromosomes, the chromosome pairs, what we call diploid, right? DI, a reference to do, diploid. Whereas in meiosis, because there is a double split, the diploid number is lost, and so the resulting cells are 4 instead of just 2, and each one of the daughter cells has a single set of chromosomes, no duplicates. In the case of the human, only 23 chromosomes. They're called haploid. The HA prefix, a reference to single, ha, single in Latin. Okay, so what happens here is that duplication does occur in the first half of meiosis. Duplication does occur, replication does occur. So this first stage, this meiosis 1, The resulting daughter cells of meiosis 1 will have 46 chromosomes each because replication did occur. But then in the second split in meiosis 2, there's no more duplication. And so those 46 chromosomes are going to be distributed evenly, 23 and 23. So meiosis 2 is the one that does the chromatic reduction itself, okay? It's very chemical, it's very mechanical, and there is an explanation for this process so that there's no magic or really not even a mystery involved here. The mystery is going to be in the creation of a soul, but not in the biological process. All right, so let's look at a little bit more in detail, concentrating on the chromosomes through meiosis, okay? So again, the cell and interphase, during synthesis, during the S phase of that interphase, the cell will duplicate, the chromosomes will replicate, standard, the same. These are the precursor cells of the gametes, okay? These are the precursor cells of the gametes. So if you want to place them functionally, for the women, we're talking about the precursors, the follicles in the ovaries. For men, we're talking about the precursor cells in the vesicular tubules within the testes. So there's replication. Now we have the sister chromatids, and here we are now undergoing meiosis, meiosis 1 and meiosis 2. Okay, so the first split, meiosis 1. Notice it's very diagrammatic, but here the chromosomes are lining up in pairs, not singly, but the chromosomes are lining up in pairs. All right, so you see, again, we have 4 chromosomes, 2 pairs of chromosome, 1 long pair, 1 short pair. So this could be 1A, this could be 1B, this is 2A, and this is 2B. All right? Here are the sister chromatids. You see that the chromosomes in meiosis remain paired up, right? So 1A and 1B are paired up, and 2A and 2B are paired up. This, by the way, this structure is called a tetrad. A tetrad because it has a total of 4 sister chromatids. 4 sister chromatids, right? Remember that each chromosome after replication has 2 sister chromatids. So if the chromosomes after replication remain paired up, then you're going to have a total of 4 sister chromatids that are compatible to each other, right? Because it's the same chromosome that we're talking about, the same chromosome number 1, number 2, number 3, number 4. And because there's a total of 4 sister chromatids in this complex, it's called a tetrad. All right, so the tetrads line up at the equator, paired up. Then the split happens, and so the chromosomes migrate to the opposite pole, but they still migrate the sister chromatids paired up. You see, so the centromere has not really given way. Okay, so the sister chromatids migrate intact to the opposite poles. And that's one distinct difference between meiosis and mitosis. There's a second one. So that the end result after meiosis 1, when we end up with 2 daughter cells, the nuclear membrane is resynthesized for each of the 2 daughter cells. You notice that inside each one of those 2 nuclei, we have sister chromatids together. All right? So really, we still have chromosome pair 1 and chromosome pair 2, and chromosome pair 1 and chromosome pair 2 also in the other cell. So and that is because during the migration to the equator, the chromosome pairs migrate together, not singly, and so that when they split up, the 2 sister chromatids go to opposite pole of each one of the pairs. So what happens is the pairs are split up, but the sister chromatids remain together. So that when the cell enters meiosis 2, you know that this process is separated like this, is fragmented for the purpose of study, but that happens in a continuum, all right? It happens in a continuum. All right, so when we call it meiosis 2, now here's one daughter cell and here's the other daughter cell. You see that it has chromosome pair 1 and chromosome pair 2 and the same chromosome pair 1 and chromosome pair 2, okay? Now what we have in the second meiosis, it is essentially a mitosis. It is essentially a mitosis. So that now the chromosome, the sister chromatids migrate to the equator singly. You see them migrating here singly. So here is, can't tell now the size. I think that this red one is chromosome 1. And the blue one is chromosome 2. And on the other sister chromatid is the opposite color. So the blue is now going to be 1 and the red is going to be 2. Okay, it's hard to tell the difference, but this is the large chromosome, this is a small chromosome. Here, this one is the large and this one is the small. So you see how they're lined up singly, but the sister chromatids are together, just like in regular mitosis. And now what gives way is the centromere, so that now each individual chromatid finally migrates to the opposite pole of the cell on both daughter cells, with the result of 4 granddaughter cells when the final split happens. And each one of those 4 daughter cells has a single set of chromosomes, single set. And you can see in this daughter cell, there is chromosome 1 and there is chromosome 2. The next daughter cell or granddaughter cell, I'm calling them because they're 2 generations. Chromosome 1, chromosome 2, chromosome 1, chromosome 2, chromosome 1, chromosome 2, and it's a total of 4, all right? And because there are no duplicates in any one of these 4 nuclei, there are no duplicate chromosomes, it's a single set. Then they're called haploid. Again, the prefix HA, a reference to single. And the number is N number. It's the N number, right? Okay, so this is in a nutshell what is happening in the ovaries and in the testicles of women and men during the production of gametes, during the production of egg or sperm. Functionally, we call that maturation, reproductive maturation, menarche, or the first time that the woman ovulates, has her period. For the man, the first time that he is able to produce functional semen that is capable of fertilizing, okay? Which is called potency. There's a further detail here that you may notice. This little color change, all right, it's more visible here. For example, in this chromosome, red has a little blue tip, and this blue chromosome has a little red tip. That is a process called recombination, and that recombination, which is also known as crossing over, recombination or crossing over, happens at the beginning precisely at the tetrad stage, at the tetrad. So the reason for tetrads is recombination. Recombination happens at the tetrad stage when the chromosomes are still in pairs, 4 sister chromatids linked together or paired up, and they're going to switch. Some are going to switch some segments from one sister chromatid to the other. Is the crossing over event. That is one of the key advantages, one of the 2 advantages of sexual reproduction, which allows for variation, right? And this is where dominant genes and recessive genes are interchanged and so forth. And so this is one source of variation, which is the payoff for sexual reproduction being on average about twice as expensive as asexual reproduction, energetically speaking, kilocalories, all right? So Sexual reproduction involves more energy, more effort, more kilocalories, about twice as many kilocalories are spent in sexual reproduction than in asexual reproduction, okay? And there has to be a payoff for that expense because nature, remember, is economic, is always trying to save energy. Unless an individual organism has to move, it will not move. All right, but they have to move to feed, typically. So the payoff is variation, this crossing over, this recombination. At this level, and then there's another level, which is, so this one is, excuse me, at the molecular level, at the chromosomal level, and the other recombination that occurs, or mixing, if you will, allowing for variation, is going to be at the cellular level, the particular sperm that fertilizes a particular egg of all the possible egg and sperm that could have been fertilized in that cycle. Okay? But that's more at the cellular level. And it's mostly from the male part because of the overabundance of sperm in contrast to a single egg in each cycle. All right, so here's a little bit of detail of the recombination process. Let's say this chromosome, we're only looking, how many chromosome pairs are we looking at here? 2 chromosome pairs, okay? Now, so this, let's say that this is chromosome 1, chromosome 1A and chromosome 1B. Okay, so these are chromosome pairs. In chromosome 1A, how many sister chromatids? 2. 2 sister chromatids, okay? 2 sister chromatids. Here's one sister chromatid, here's the other sister chromatid, all right? For chromosome 1B, the same argument. 2 sister chromatids, 1 sister chromatid, and another sister chromatid. So when we see a diagram like this, we already know that replication has occurred. Replication has occurred. So there's a temporary doubling up of the genetic material. Okay, and each sister chromatid also has 2 arms. That's why I mean there's a lot of 2s here, all right? 2 arms, and typically the centromere is more or less in the center, but it's not exactly in the center. It's kind of off center, slightly off center. So we talk about the long arm and we talk about the short arm, the P and the Q arms. It's a little bit too much detail, but that's just the genetic classification or the genetic nomenclature. Okay, so sister chromatids, each sister chromatid has 2 arms, a Q arm and a P arm to try to get down to where the genes are. All right, so this crossing over or the lining up, the tetrad formation is also called synapsis. And here is the actual crossing over. It's just shown in a caricature. There's a very elaborate biochemical process that happens here. A bunch of proteins are involved, protein complexes that have to nick and cut one segment of the ladder, of the DNA ladder, and connect it with the other segment of the ladder that was also nicked. And then that has to be sealed over with different protein complexes, all right? And the end result is that chromosome 1A got a piece of chromosome 1B, and chromosome 1B got a piece of chromosome 1A, complementary pieces, because we expect the same type of genes to be in this area. This would be like the distal end of the Q arm of sister chromatid, of sister chromatids. But we have like equivalent genes here. The only difference at the very genetic level is that maybe the dominant genes are in the blue and the recessive genes are in the brown or the red. So when we do this flip over, the recombinant chromatid blue lost the dominant gene here for brown eyes and picked up the recessive gene from the red sister chromatid of blue eyes. So that's a recombinant. It's the same gene generally that determines, let's say hypothetically, the proteins that determine the color of eye in the iris. But there's a dominant and there's a recessive, and they got flipped over. That's recombination. Okay, so this is recombination. You can see functionally that this allows for variation of the progeny, but still maintaining the same species functional. That can work. So it's mostly about sharing dominant and recessive traits, but always of the same gene because they are complementary chromatids. In other words, for example, chromosome 1 will not line up with chromosome 2, will not form a tetrad with chromosome 2. And chromosome 5 will only form a tetrad with its complementary chromosome 5 and not with chromosome 23. Okay? And further, the P arms will only pair up with the P arms and the Q arms will only pair up with the Q arms. So the tetrad is a very rigorous structure, very rigorous. Okay, now just flip that thing over so that we can see it in the diagram. Here's the recombination process. And notice that of the sister chromatids, only 2 sister chromatids recombine, but other sister chromatids do not recombine. So it's at the level of individual sister chromatids that recombine or not, so that we can talk about recombinant chromatids and non-recombinant chromatids. At the end of the day, recombinant chromatids and non-recombinant chromatids are going to end up in different cells, in different gametes. So that some gametes will be recombinant gametes and other gametes will not be recombinant gametes. For each individual gene, and we have an estimated between 10 to 20,000 genes. So you can imagine just by this simple, elegant process of recombination possibility for each of the 20,000 genes, the amount of variation that can be introduced into the next generation on each particular trait, okay? On each particular trait, and there it is. So this is all determined by the proteins that work, do the working of the recombination work, but those proteins themselves are being coded by the DNA. So the information continues to be in the DNA down to the level of coding the recombination process. All right, let's back out a little bit now, go to the cellular level. At the cellular level, meiosis is the formation of gametes, which are the sex cells, and genetically they are haploid. Understand that? This process of meiosis is actually called gametogenesis because it's the formation of gametes. Again, we like to use sophisticated words. So genesis, a reference to formation or generation of gametes, gametogenesis, which in the male is spermatogenesis because it's producing sperm, and in the female is oogenesis because it's producing ova, one ovum, several ova. So the gametes, the sperm and the eggs are always haploid in number. For the human, it is 23. Okay. I think this is more the same. All right, so here now I'm focusing on the cells themselves, on the gametes, right? So these are the precursor cells, which are going to be, they're called primary spermatocyte for the male, primary oocyte for the female. In general, this is for mammals in general, okay? These spermatocyte and these oocyte, are they haploid or diploid? They're 2N. They have chromosome pairs, so they're diploid, okay? These are the precursor cells. These are within the ovaries for women, within the testicles, in the vesicles, in the tubules, in the male. So these are the precursor cells, but they are diploid themselves. They are diploids, they're precursor cells. They undergo the double meiosis, and we end up with granddaughter cells, I'm calling them. There's a difference. There is a difference between male and female in this. In the male, we end up with 4 sperm, 4 gametes, essentially identical to each other. except for the genetic mix, right? The recombination that may have occurred in each one of those 4 gametes, but these 4 sperm are essentially identical to each other. And in principle, the 4 are functional. In other words, capable of fertilizing, all right? So from 1 spermatocyte diploid, we get 4 sperm haploid, essentially identical to each other functionally. In contrast, for the female, from each oocyte, we only get 1 egg, 1 mature functional egg, 1 ovum. However, there are really 4 cells that are produced, 4 gametes, because it's a double split. So we have to start with 4 daughter cells. We have to end up initially with 4 daughter cells. However, notice here the difference. At the first split, first I was concentrating on what was happening to the chromosomes. Now I'm going to concentrate on what happens to the cytoplasm. For spermatogenesis, the splits are even splits of cytoplasm, even split of cytoplasm, so that the mother cell will split into 2 daughter cells of even amount of cytoplasm, even volume of cytoplasm in the 2 daughter cells. Each one of those 2 daughter cells splits again with an equal amount of cytoplasm, with an even amount of cytoplasm, so that the sperm end up with an even amount of very little cytoplasm, mostly nucleus, but they're all the same size, the same shape. In contrast, at the first split of the oocyte, there is an uneven distribution of cytoplasm, uneven distribution, so that one of the daughter cells has most of the cytoplasm, one of the daughter cells, and the other daughter cells has very little cytoplasm. It's already becoming degenerate, non-functional. So that's the difference, and it's at the cytoplasmic level. Again, directed by the proteins that do the work, all right? So that the pinching of that first split in the oocyte is not evenly along the equator, is disproportionate, is off to toward one of the poles or the other, all right? So that one of the 2 daughter cells ends up with most of the cytoplasm and the other one little. The second split happens because it has to happen. You know, this is it's a one-way automatic thing. The second split happens in both of those cells, but the same thing happens again. In the cell that has most of the cytoplasm, one of the cells ends up again with most of the cytoplasm and the other one with less, and also becomes degenerate. So that at the end, we end up with 1 functional egg, ovum, with most of the cytoplasm, and the other 3 with very little cytoplasms. They're degenerate, and they're called the polar bodies. Okay, the polar bodies. And the polar bodies just kind of hang around. They get stuck here because this X also is shown in a simple way, but the reality is that this egg is actually a complex. As we know, it has 2 layers. It has a layer of the zona pellucida, and then it has another layer of cells, the corona radiata. So this is actually a complex of cells functionally. And embedded there in the zona pellucida are the polar bodies that are just hanging around for the ride, but they can't get fertilized because they don't have enough cytoplasm to sustain a fertilization. So again, there is a very detailed explanation of this process that we know now, at least at this level, and therefore there's really no magic or even mystery here at the organic level, okay? This is kind of a summary for our species. The total number of chromosomes is 46 or 23 pairs, but technically speaking, these 23 pairs are only identical, all 23 pairs are only identical in the female because the sex chromosomes for the female are both X, so XX. But in the male, because the sex chromosomes are XY, they're not strictly identical. The X is not identical to the Y and vice versa, okay? Because it's missing one of the legs, it's actually a little shorter, the Y chromosome, which determines malehood. All right, so we can talk about, of the 23 pairs, 22 of those pairs, there was a vast majority of them, meaning also the vast majority of genes, are autosomes. Autosomes, they're involved in things other than reproduction, than coding for the reproductive organs. One pair of chromosomes has the task of coding for the reproductive organs, and those are the sex chromosomes, and it's one pair, and it's typically the last pair, all right? Not because they're smaller, they're actually larger, but it's just the last pair. So pair number 23 is a sex chromosome, and that's what they're called. They're called sex chromosomes. The other non-sex chromosomes are just called autosomes. Now, let's back out further to the level of the glands, of the reproductive glands that produce these cells. Okay, where does gametogenesis occur? In the women, it occurs in the ovaries. Oogenesis. In males, it occurs in the testes or testicles by spermatogenesis. The precursor cells are embedded in follicles for the women, the ovarian follicles, all right, which mature on average once a month, and it's a lunar month. In theory, 28 days has to do with the pull of the moon somehow, with the gravitational pull of the moon on the water of the body of the female, opposite to the gravitational pull of the earth. That somehow has a chemical effect on mammals, okay, on female mammals, so that it produces a cycling. But this cycling is arrested because you notice that it undergoes the primary oocytes, which are still diploid. The follicle matures, and it goes all the way down to the first meiotic division. And here, the cycle is arrested. It does not go into meiosis 2. Does not go into meiosis 2. These precursor cells are already haploid, but they need to split. Okay. In the male, the area where the precursor cells are the seminiferous tubules inside the testicles. These are the primary spermatocytes. They are diploid. They split, meiosis 1. There's a haploidy. They split again. So we end up with 4 functional chromosomes, 4 functional sperm on average. Okay. The polar bodies are not shown here, but they're embedded around the cumulus somewhere. Oh, okay, here, first polar body. All right, let's talk a little bit about the reproduction functionally or maturity, sexual maturity. A species will have basically, as far as development is concerned, we'll have 3 stages of development. Generally, we have the embryonic development from fertilization to birth. Talking about mammals, talking about humans as mammals. So the first stage will be the embryonic stage, will be from fertilization to birth. Then the juvenile stage, right? Juvenile stage until sexual maturity. Once we reach sexual maturity, then functionally we become adults. Organically, biologically, functionally, physiologically become adults, even though psychologically we may be very far away from adulthood, all right? Because of this phenomenon of adolescence that is fairly relatively new in our history. Why? Because early on, for many centuries and millennia, the kids had to go to work and they matured quickly. And they even died while they were working and everything else. So there was practically no luxury of adolescence. And when they hit sexual maturity, they also hit psychological maturity. And we're talking about early teens, 10, 12, 13, it varies. Some boys and girls may mature as early as maybe 8 or 9, in part, depending on what kind of hormones they were exposed to also in today's diet. But let's say at 10 years of age on average, okay? Just to choose a round number. At 10 years of age, a young boy, if he's able to produce semen that has sperm, then he's reached biological maturity, okay? And those sperm in principle are capable of fertilizing. 10 years of age. For women, what happens is that the eggs are arrested at this stage and the final development will happen at 10 years of age, meiosis 2. And that is what is called menarche, or the first period, the first ovulation, okay? Now, in contrast to men then that are producing sperm essentially from the first production of sperm until we die, our testicles are producing sperm all the time, 24/7, okay? But the sperm itself only lasts about 24 hours and then degenerates. So what happens to all those millions of sperm that we're producing constantly? Why don't we just blow up? Because they die, they become degenerate, and these seminiferous tubules and the vesicles that are outside, the vas deferens and all the vesicles that are in the scrotum reabsorb the old sperm, which is 24 hours old, okay, is being reabsorbed. So constantly there is a replenishing, there's a reabsorption of old sperm, and there's a production of new sperm. So in principle, every man has potent sperm available for fertilization, biologically speaking, and then when ejaculation happens, millions of those sperm are released in the ejaculate, and there's a temporary down emptying or partial emptying of the tank, if you will, of the reservoir sperm, but within 24 hours it's being replenished again, all right? To finish that story, do you know how many sperm are in a functional ejaculate to be considered potent, to be considered biologically, genetically potent or capable, capacitated, capable of fertilizing? There has to be clinically between 100 to 150 million sperm for an ejaculate to be considered potent or capable of fertilizing, okay? And yet, how many sperm does it really take to fertilize one egg? One sperm. And so you can see this is natural selection. This is selective pressure. The selective pressure against human fertilization is about 100 million to 1. 100 million. So what does that mean? That on average, because we're going into a probabilistic model here, the sperm that gets to fertilize that egg, that generated us, for example, all right, is on average the most potent, the most capable, the most capacitated sperm, the most vigorous sperm, you know, cellularly and genetically to get there to fertilize that egg, because within a nanosecond later, there's another sperm that's coming right behind them. Okay? And another one to the tune of 100 million. Well, not all 100 million make it all the way up there because they have a lot of barriers on the way, especially the crevices in the endometrium, right? Which in the meantime has also thickened to prepare for a possible implantation. So these are road obstacles, if you will, for the sperm. And that's why competency has to do with the wiggling, with the motility of the sperm. All right, so there's a tremendous, immense selective pressure against sperm in mammals in general and in all males in general on the reproductive process. That's why on average, most of us are kind of normal when we're born, because the abnormal got selected out. Either they never fertilized, or if they did get to fertilize, those abnormalities were not compatible with life. In other words, they were discarded, typically in the first couple of weeks of the embryonic development, and it's called a spontaneous abortion. If it's later in the pregnancy, then it's called a miscarriage. But it's a natural event of selecting out those who were not genetically fit, really. They were not compatible with life, okay? So in contrast to the maturation story of the sperm, which happens once these cells are ready to mature, on average about 10 to 12 years after birth, with women, the absorption, the reabsorption occurs during those first 10 years. So a woman is born with a set number of primary oocytes, okay, a set number, which is going to be in the millions. By the time she hits menarche, an average of 10 to 12 years later, she only has hundreds of thousands of eggs. So she has lost the vast majority, 99% of her eggs have been lost. Why? They became too old. They're 10 years old. They became very too old and were reabsorbed. So what happened to those old eggs? They got reabsorbed by the tissue, by the ovary tissue, okay? And nature is economic also this way. It recycles, it breaks down the cells, and reabsorbs the nutrients for other cells that are around. So by the time she hits menarche, she only has hundreds of thousands or maybe tens of thousands of eggs, which is still an incredible excess of eggs. Why? Look at the math, look at the arithmetic, very simple. Let's say that a woman is fertile from 10 to, you want to put it at 40, you want to put it at 50. Let's put menopause at 50, which is kind of late, right? Okay, so we're talking about 40 years of fertility, from 10 to 50, 40 years. 1 egg a month, so how many in a year? 12 eggs. She needs 12 eggs a year to be fertile every month of the year. 1 year, so 12 eggs times 40 is 480, right? 48, 480. She needs less than 500 eggs to be fertile every month of her life for 40 years. Okay? Less than 500 eggs. And she's got tens of thousands. So again, those eggs are going to continue to degenerate until she really does run out. And then menopause. She runs out of the last egg, which at this point is now 40 or 50 years old. It's incredibly old. And I'm making an emphasis on this because genetically, old means more possibility of mutations and chromosomal abnormalities, and also in the meiotic process, particularly a process that is called aneuploidy, which is that the sister chromatids don't fully separate at that meiosis 1 or meiosis 2, and they stay stuck together, and that's how we get the trisomies, for example. Trisomy 21, which is the most common one, is the only one. The trisomies happen really with all the chromosomes, but for most of the other chromosomes, they have more genetic information, they each carry more genes, and therefore it's not compatible with life, and those are spontaneously aborted. Those embryos are spontaneously aborted. Typically, like I said, within the first 2 weeks of the pregnancy, the woman doesn't even know she's pregnant, she just thinks she's late in her period, she's having actually a spontaneous abortion, okay? And so, with chromosome 21, which actually is the smallest chromosome, and happens to have the least genetic load, in other words, the least number of genes, then when there's an extra load of that chromosome, it is still compatible with life. But it carries a set of abnormalities, which is called a syndrome, all right? And the syndrome is both at the physiological level and or the psychological level. And there's a range of those syndromes at both levels, at the physiological level and at the psychological level. At the physiological level, one of the typical characteristics is the little slanted eyes. It used to be called Mongolism because it looks a little bit like Oriental eyes, right? And also the twisted tongue and somewhat the mouth formation also somewhat abnormal. That's at the physiological, which could be from the most severe to the most slight, almost not noticeable. Psychologically also, retardation, mental retardation from the slightest to the most severe in a range. So you combine those two, and those are your Down syndrome children, which again is a range on the both levels, but it's just about there are others, sometimes chromosome 18, there are Patau syndrome and other syndromes. Sometimes fragments of DNA get stuck precisely at the recombination level, and it was not totally clean, it got stuck and it got carried with it, with one of the sister chromatids. So all those are genetic abnormalities, and they tend to happen later in life. And that's why on average, from about 32 years of age, correct me please if I'm wrong, but between 32 to 35 years of age is when Down syndrome trisomy just increases geometrically, all right, exponentially. And so it's a big difference for a woman to have her child before 32 than after 32. She's much a higher risk of having a Down syndrome child or any other abnormality definitely after the mid-30s. This compounds a problem now with women having careers, and then they want to, of course, once they get the degree, because they really sweated blood, sweat and tears for that bachelor's, and now they want to put it to work, so they develop a professional life, and the children are going to be in the way, so they postpone having their children until their 30s. And then the result is what it is. So it's a big challenge, and frankly, it's a dilemma for me. I see a difference between a 20-year-old man starting a career and a 20-year-old woman starting a career. And I see that there are going to be more challenges for the woman on average, especially if she wants to have a family. So these are the issues that we have to deal with today. But that's a reality. And it's the truth. And there you go. And there you go. Okay? And these are the challenges. Yes?
What about for those individuals that are blind or that have hearing and they want an offspring? Is that passed on to their
It may or may not be. It has to be if it's congenital, if it's at the genetic level, or if it's something that they acquired later in life, or even in the embryonic stage. But it could be some kind of physiological abnormality that is not necessarily at the genetic level. So we really have to go for every single abnormality that we see in the human, whether it's physical or mental. We have to study it individually and see if it's an inherited characteristic or if it was acquired during the lifetime, even the embryonic lifetime of the individual. Okay, so really the answer is it depends. It's like when in medicine you say you see a patient, you've seen a patient, one patient. And many patients may have a similar disease or the same disease, but it's still patient by patient. Okay, all right. To conclude here, because now after this I'm going to go into the fertilization process itself, which is a good time for break. So let me show you this little video, which actually has the development, embryonic development from fertilization to birth in 6 minutes. All right, and it's very well done. So of course it shows the highlights, but I think it's extremely well done. So let's take a look at it for a moment, and then we can break. I'm going to mute the sound because for some reason this author chose crazy music, some kind of rock thing that is not conducive.
This is a Devoted Health Medicare plan. Actually, it's a Medicare HMO plan.
It also
Devoted Health takes all the benefits.
It also mutes the apps. Okay. So the music is not relevant and there's no explanation, so I can go a little bit. But it's a couple, and apparently the husband is very savvy, technically speaking. Here what we see is ejaculation has just happened, and here we are in a mass of a sperm traveling through the endometrium of the vagina. Is actually going to go through the cervix and into the fallopian tube. Okay, so you can see the endometrium here, the lining up, right? And all the crevices, the crypta, as these sperm have to navigate through all that. Many millions get stuck there because they're not as competent, they're not as strong, as capacitated. And so already the losers are being left behind. We can see at the proximal end here of the uterus above, the two openings of the fallopian tubes. And we know, of course, that at that point, another 50% of the sperm are going to be eliminated from the race because, you know, sperm comes from the men, and we don't ask for directions. So half of that sperm is going to go into the wrong tube. Exactly. You're going to go into the wrong tube. There's no egg to fertilize there. The egg fertilizes over here. So we lost another 50% just by chance, flipping a coin. Okay. All right, it will continue then, and it'll start focusing on an individual sperm at this point, a little further. Okay, there it is. In the meantime, what we have is we have the cervical mucus that is flowing downward, and that cervical mucus also has changed biochemically to become hospitable for the sperm. So it actually maintains the sperm alive up to 5 days, after the ejaculation itself. Okay, there is the egg with the cumulus, is the complex structure. Outside is the corona radiata, then there is another layer called the zona pellucida, and the egg has to penetrate all that. The tip of the sperm has digestive enzymes there. They're being poured out, the digestive enzymes. That's called the acrosome, the tip of the sperm. I'll show you the diagram in a minute, but basically that was the pouring out of digestive enzymes that allow the sperm to literally bore through the corona and then the zona and finally get to the cellular membrane of the egg and empty its nucleus, its haploid nucleus, called a pronucleus, into the egg itself. Okay. So you see motility is used up to the very end. The second sperm just got left out. There is a process called the cortical reaction that seals the egg from allowing the second sperm to fertilize. Okay, now we see what has happened is the head of the sperm has entered. Now we are in the cytoplasm of the egg. The tail has been lost outside. And here now the membrane of the sperm is being digested away. And what is going to be left is just the pronucleus, the haploid nucleus of the sperm, which will then need to fuse with the haploid nucleus of the egg, the other pronucleus, to restore diploidy, So there's the male pronucleus, the female pronucleus, they fuse, and that's the second level of fusing, and then we just saw mitosis occur, the first mitosis, all right? That took about 24 hours. And now, from here on is mitosis until they get bored, and then do some more mitosis for X number of years until this individual is going to die. Okay? So this first cell, which is the product of fusion, fertilization, is called the zygote. We'll get to it in the second part. And that's the beginning of a new individual there. Now we see that this is the chromatin inside that zygote is beginning to split. Now here is replication. All right, this is obviously a diagram. These are not photographs, not yet, but there is the replication. The sister chromatids lining up, and they're going to line up singly because this is mitosis, and we just saw mitosis occur. The two daughter cells are forming now. All right, and that's the first mitosis within 24 hours of fertilization. Now the egg continues to be inside, sorry, not the egg, the zygote continues to be inside that cumulus cloud of zona and corona radiata. And at some point, just before implantation, there's going to be a hatching. The embryo is literally going to come out of that soft shell, all right? Reminiscent probably of our reptilian ancestors. All right, now the egg is falling, the embryo is falling down out of the fallopian tube to the endometrium, right, into the uterus. And watch, now the embryo has undergone 2 more stages from the zygote. As that zygote is splitting, duplicating the cells by mitosis, is going to form the second stage that is called the morula. And then the morula will continue to split and start differentiating into 2 different types of cells, forming the blastocyst. The blastocyst that will implant 5 to 7 days after fertilization in the human. And so before implantation, that little blastocyst, or little embryo, actually has to come out of the cumulus of the shell, of the soft shell, and that is a type of hatching. You will see. There we go. So, okay, now we have the actual embryo that is going to start burrowing into the endometrium that has been enlarged and thickened, and that is the implantation process. Now, he's focusing on the development of the fourth stage of the embryonic development, which is called gastrulation, which is the embryo starts becoming oblong, gastrulation, and now the gastrula will eventually form the three germ layers, the ectoderm, mesoderm, and endoderm, which are the three germ layers that form all the organs of our body, okay? And there's an extension of the embryo, a lengthening of the embryo to a head and a tail end, all right? So there'll be a head and a tail end, and a groove in the middle that will pour in cells called invagination to form the nervous system. Okay, that's going down the groove from tail to head, and you see that that fold then closes over, and the beginning of the vertebrae, there's the beginning of the heart, the absorption of the remaining yolk. We develop a tail bud temporarily, reminiscent, vestigial of our tail ancestors, and that tail is reabsorbed. In the meantime, the placenta is being established into the endometrium of the mother, into the uterus of the mother, and development continues. Cephalization, the head, the limb buds appear, the forelimbs, in other words, the arms, and then the legs, and then digitalization as the cells continue to develop and specialize. All of the organs of the head start appearing, and the internal organs also. By the time the embryo is 8 weeks old, 2 months, that embryo has all of the organs and systems in the body in place, the major organs and systems, but in miniature. And technically, the embryo changes name from fetus. So the next 7 months is just growth in size, mostly growth in size. But most of the organs and systems are already formed by the second month, by 8 weeks. So the fetus is moving around, sucking its thumb and making the mom crazy, and opening and closing their eyes, playing with the umbilical cord. What is this thing? They suck their thumb, they suck their toes, they want to get out of there. They can see some light, they open and close their eyes. They're peeing into the amniotic fluid because that's how they're getting rid of the toxins, of the urea. But they're still in a liquid environment. So we spend 9 months in a liquid environment. And that's why it's very good to teach babies how to swim early on, because they're still connected to that liquid environment. So there's mom about to give birth, and the dad who did all the filming and editing, and there's Junior 9 months later. Okay? I think it's a little baby girl. Yeah. Hi to the world. Get ready. Your first bath. Amazing, huh? All in 9 months, and that is the fastest we grow in all our lifetime. I did a little arithmetic one time, so that the zygote, which is about 10 microns in diameter, to a baby, which is an average of about a foot more or less, the growth in sheer size is about 5,000 times, all right? From the fertilized egg, from the zygote to the time we're born, in less than a year, we grow 5,000 times. It's by far the fastest time that we grow, and all the development that occurs, critical. And that's why for mammals, gestation is internal, because trying to protect that embryo, that fetus, from all the insults that will occur on that human being for the rest of our lives. Once we're born, let's say we live 80 years old, which is relatively long. I don't care, you can put it 100. We will grow, what is a tall person? 6 feet is a tall person. So for the next 80 years, we grow a maximum of 6 times more. Okay, but for the first 9 months, we grew 5,000 times. And then the next 80 years, we grow maybe 6 times more, and that's about it. And even the bulk of those 6 times is done in the first 10 to 20 years, right? Okay, so that was very fast. We're gonna go back to that in a little more detail, slowly but surely, as we look into fertilization, all establishing in luxury of detail, biological, physiological, natural detail on when that new individual begins. Any question or comment? No? Are we good for a break? Let's see, there's a little expansion here. Okay, we're going to take a little break now, if you can hear me, I hope. Maybe 10, 15 minutes, and we'll be back. I'm going to pause this. Okay, see you in a little bit. Okay, we're resuming recording. I'm just looking at the handout for a moment, which is 2 sides. On the one side, the front side, if you will, it has a summary, the keywords of what I covered with regards to meiosis. Okay, and the big event functionally for meiosis is this chromatic reduction, reducing the number of chromosomes from diploid to haploid, so that the gametes are haploids, and those are the ones involved in fertilization. Because then fertilization, if you will, reverses meiosis, in other words, restores diploidy. Okay, so functionally, fertilization is the reverse, by the gametes coming together. Then we end up with the diploid number of chromosomes, and that is the most logical place where the new individual begins, because you can see that the egg and the sperm are part of the parents. In other words, the egg belongs to the woman, the sperm belongs to the man. There are cells, there are specialized, very specialized cells of those 2 individuals. But once those 2 gametes fuse, now we have a new entity, a new biological entity, which is no longer haploid, it's diploid. It is no longer 100% the woman, nor 100% the man. It has 50% of the genetic material of each. Okay? And so it is truly a new individual altogether, of the same species for sure. And then from that point on, it's just mitosis, is cellular specialization, differentiation that is called. And that's what we're going to look at now in the early part of the embryonic development. So I'm flipping over to the back side of the handout. Okay.
Are you recording, Carlos?
Yes, thank you. Yes. There's a little red dot there. Okay. Thanks. Okay, so let me dim the lights here. Let the eyes adjust for a moment for the note taking, if you wish. All right, the zygote, remember there's an added value to this program in that you pick up a little Greek and Latin on the way. And zygote comes from the Greek zygon, and zygon means yoked or linked. Okay, yolk or linked. Not the yolk of the yolk sac, because that has an L here, all right? Yolk is like the yoke of oxen, of 2 oxen, that piece of wood that is like 2 U shapes that unites 2 oxen when they're plowing the field, for example, okay? That's a yoke. It unites or linked. That's what it means in Greek. And the linking is at least at 3 levels, There is the linking or the pairing up, if you will, at the cellular level with the egg and the sperm fusing. The actual fertilization is the egg and the cell fusing is the first level of linking or fusion. The second level is the 2 pronuclei also fusing, all right? And the third level is actually the pairing up of the chromosomes, particularly when they replicate. That is the third level, and that is the final level, if you will, of the fertilization process itself, which is called syngamy, and I'll get to that in a moment. All right, so those are 3 levels of yoking or linking or coming together, pairing up, at the cellular level, at the nuclear level, and at the chromosomal level. In the zygote, 50% of the genetic material is from the mother and the other 50 from the father. So we have a genetically new individual here. And that's where it starts, by defining the new organism. Okay, let's look at egg and sperm a little bit closer. Here is the egg with the nucleus, which is actually called a pronucleus because it is haploid. So it's a pronucleus, PRO. Then the cytoplasm, of course, and the zona, you see the zona pellucida, and then the corona radiata, all right, cumulus of cells. Also, these little vesicles here, shown in blue, these are cortical vesicles or cortical granules. They are full of calcium and they are responsible for causing that cortical reaction. In other words, when the sperm connects here all the way at the level of the corona, these vesicles release the calcium, and the end result of that calcium release is that the egg seals. It seals, it's called a cortical reaction, and it seals the egg so that no other sperm may penetrate. I find it interesting, and that is it's a calcium imbalance reaction because it is also calcium that calcinates the egg in birds and reptiles and makes it actually hard, literally hard, right? But there's a sealing also of the egg there that calcium is involved in that metabolism, that process. Okay, and so the egg, it tends to be round with quite a bit of cytoplasm because it is we'll put it in location in a minute in the ampulla of the fallopian tube, but structurally we see that the sperm is very different. Just about the only thing that is equivalent to the egg in the sperm is the nucleus, which is also a pronucleus, but it is a male pronucleus, okay? It's a male pronucleus. And let me just move forward here for a moment, see if I have it. No. I forgot to mention, just very briefly, which I know you know, but let's go back here for a moment because I forgot to mention that sex determination is done in the male at the sperm level in the first meiotic split because in the female, the 23rd chromosome is an identical pair, right? It's XX because the female only has X chromosomes in her, in every cell of the body, including the oocytes. So when that primary oocyte splits and becomes haploid, each daughter cell can only receive an X chromosome. So 23X and 23X. In the male, however, because we have the uneven 23rd pair, XY, okay, XY, it just means that the Y chromosome is a little smaller. It's actually disappearing in evolution because it essentially just holds the SRI gene, the genes that determine maleness, that code for differentiating the gonads into testicles instead of ovaries, okay? I'll get to that in a minute. But basically what I want to say is that the 23rd pair of chromosomes in men is uneven, XY, and so when the first split happens, the first meiosis happens in the distribution of chromatids, then one set of cells will get an X chromosome and the other set will get a Y chromosome. And that follows through in the second split, so that in the end of those 100 million sperm in every ejaculate, on average, 50% will have an X chromosome and 50% will have a Y chromosome, on average. Okay? Therefore, on average, again, we're going to a statistical probabilistic model, on average, the sperm that fertilizes the egg, half the time is going to be an X sperm. And the other half of the time, in any given fertilization, is going to be a Y sperm. It's not that they alternate, because we know that couples, sometimes they have 4 or 5 girls in a row, then they have 2 boys in a row, etc. Okay? But in the population, we're jumping to a population biology here. on average, 50% of the progeny are going to be boys and 50% are going to be girls. And that's a good thing because that continues to maintain the even ratio of boy or girl. In fact, functionally, we have slightly higher. I think it's about 50.5% women and 49.5% men in the US. But it's within 1% of difference, which is negligible. Okay, so on average, every woman will find a mate and every man will find a mate, in society at the population level. That's one of the big dangers when certain governments or regimes start doing population control and eugenics and social engineering, as in the case of China, for example, which is known. They have this one-child quota on couples. What happens? Most couples, naturally, they prefer firstborn boy. And also for economic reasons, because it's more expensive to raise a girl, to the point that in some countries, to get the girl to marry, the parents have to pay a dowry, have to pay money to get the girl out of the house to marry a man. And so injustices that exist, culturally, that's the way it is. Therefore, what happens, there is an active eugenics movement of killing unborn girls in China and other places of the world because of this preference. If I can only have one, then I want it to be a man. Also because the male can work and more and be stronger and everything else and produce more for the house. So it's very materialistic. But now it has come back to haunt them because by doing that for decades, they have skewed the population to where there is an excess of males, statistically significant, larger number of males. And they are competing for these females, literally, physically, all the way to fist fights and fights, like also because everybody works for the government in these factories. And why is everything made in China? Because it's very cheap over there. Labor, very cheap, And so in factories where they have men and women working together, you have all the men fighting for the women, literally, physically. And it's a disaster because they have skewed the population, and that will continue to go on for those whole generations until, they die and the new generation comes and replaces them. So now, because of that, China has now, in certain parts, apparently allowed for couples to have 2 children. But still, it's anybody's guess how that's going to play out. Any kind of manipulation on the population, the human population, is extremely dangerous, and it can come back to haunt us big time. Not to mention, of course, that the actual replacement of human population is considered to be about 1.7. It's a little under than 2 for some reason, so definitely more than 1. And if we fall below 1.7 replacement, all right, so what does that mean? That on average, every couple is having 1.7 children. So you can't have 1.7 children, right? You either have 2 children or 1 children or more, but this is an average. If a country has less than that ratio, what happens is that the population is aging gradually, and then what? We get into retirement, we're drawing on Social Security, but where's the labor force? There's less and less of labor force to maintain us retired. Okay, and so the population ages, and what gets hit? The economy gets hit. And other economic problems arise, and all kinds of imbalances because we fool around with nature. And remember, nature never forgives. Nature cannot forgive because it can't. It's not a thinking process. Okay. So anyway, this is, I'm sure you know, just to remember then, on average is the male that determines, and on average is a 50-50 precisely because of the XY split at this level. Okay, so we can talk about X sperm and we can talk about Y sperm. It's still sperm, but it's a Y sperm that would generate a boy, or the X sperm would generate a girl if everything goes well. All right. So then, whether it's X or Y sperm, the nucleus is still a pronucleus. It is haploid. There is the little acrosome, the capsule where the digestive enzymes are contained at the tip of the head. The rest, it has a mixed section filled with mitochondria because these are the energy factory of the cell. And this cell is a very motile cell, right? It has to wiggle, the tail, the flagellum has to wiggle quite a bit to get all the way. If you compare the distance to size, to scale for a sperm, which is microscopic, to travel that whole distance to negotiate first getting through the cervix, and then against the stream of the cervical mucus, which is flowing downward, and through the crypta of the endometrium, and then eventually into the fallopian tubes. And there are these hairs that are moving downward also. The sperm has to negotiate all that, so it's obstacles all along the way, okay? It would be the equivalent of, I don't know, running for miles and miles and miles to a human scale, to the size of a human being. And that's why many of them actually get tired. This is an actual micrograph. This is an electron photograph, microscopic photographs that has been colorized to scale, to size. So you can see that in proportion, the egg has much more cytoplasm than the sperm, which is basically just that compacted pronucleus. And here's a scale. Is 1/10 of a millimeter, which is, a millimeter is more or less the thickness of a nail, all right, a fingernail, and that's the egg, whereas the sperm is 0.05 millimeters, so much more, it's about 20 times smaller to scale. Okay, here is it's just a schematic of the fertilization process beginning here. Number one, the first sperm making contact, releasing the digestive enzymes from the acrosome, boring through first the corona, then the zona, emptying out its pronucleus, migrating to fuse with the female pronucleus to generate the diploid nucleus, now a true nucleus diploid. And this whole process here is called syngamy, okay? Syngamy is the coming together of the two pronuclei, and when syngamy is complete, then we can say that fertilization is complete and we have truly a new human being at that process, because it's a new individual that has its full genetic material functionally ready to undergo mitosis. This little small blurb over here is one of the polar bodies that you can see it's embedded into the zona, just trapped there, okay? And this is an actual micrograph. It's an electron microscopic photograph that has been colorized. As you can see, one sperm arrived and the others were just right there, but they got locked out by that cortical reaction. So on the male side, there is an acrosome reaction, and on the female side, there's a cortical reaction. Cortical referring to the cortex or to the surface, just like the cortex of the brain. Okay, let's put the egg and the fertilization process in locus, in place. Here is the horn of the fallopian tube, and it happens precisely at the bend, which is called the ampulla, okay? That is the normal place. So you can see functionally, here's ovulation. The egg will migrate slightly up to here. Of course, this is not really to scale. This fallopian tube is been enlarged for the purpose of the diagram, and the egg also has been enlarged tremendously just for the purpose of visualization. Okay. This area called the infundibulum, this little finger-like structures, you notice that the distal end, the end of the fallopian tube, is not actually connected to the ovary. Okay, there's a little gap there, which here is exaggerated. It's just several millimeters in distance, but there's a little gap between the ovary and the infundibulum, the hand of the end of the fallopian tube. And when the woman is ovulating, that little hand, that end of the fallopian tube will actually move close into the ovary and wrap around. And that little movement causes the pain that is known as Mittelschmerz, the middle pain in German, the middle pain, which is in the middle of the period. It's not the period, okay? It's the Mittelschmerz is the actual hand moving, is the fallopian tubes moving toward the ovary to catch the mature egg. And it causes a little bit of pain. various degrees. Some women don't feel anything, other women feel a lot, but it's not the menstruation pain. It is actually in the middle of the cycle, and that's why, and it's also in the middle of the body. So the Germans who discovered this first and found out what the pain was all about, they call it middle pain, Mittelsmärz. It's one of the symptoms, it's one of the secondary symptoms of ovulation. I mention it because later when we get to natural family planning, it's one of those 7 secondary symptoms of ovulation, okay? Anyway, the egg will reside here for about 24 hours, which is its time of maturity, just like the sperm also is only 24 hours within the testicles, and then they over-ripen. If there's no fertilization, this egg is then discarded through a peristaltic movement. It's like a marble inside a hose that is being pushed on the edge, and the marble continues to move down the hose. It's that movement, it's an undular movement on the fallopian tube, which is similar to what occurs when food is moved down the intestines, okay, peristalsis. Anyway, that egg is pushed out when there's no fertilization, and when it gets to the uterus, it is discarded together with the endometrium that has been thickened in the meantime, preparing for an implantation, and now that whole tissue is sloughed off, is discarded. There's a lot of vascularization there, the capillaries are broken, and that is what is causing the bleeding in the menstruation, okay? So amid all that bleeding and some tissue will be a very tiny microscopic egg, normally, that is over-ripened. That if there is no fertilization. If there is fertilization, meaning that at least one sperm made it all the way up to here and was capable of, was potent enough to fertilize, then the process changes, because now we have a zygote, and that zygote is going to undergo mitosis and start splitting. It starts the embryonic development, okay? By the way, just to complete this picture, I said that both egg and sperm on average are mature for about 24 hours, but that's in place. In other words, the egg in its own place and the sperm in the seminal vesicles. However, the sperm inside the female tract is capable of living up to 5 days, because the cervical mucus, which is actually lined up, the whole internal reproductive system of the woman is lined up with this mucus, the cervical mucus, has several functions. One is a bactericide, it is a germicide, okay? But also it is a highway during fertility, it is a highway for the sperm to travel through the mucus, all right? Again, I'll talk about that in more detail when we get to NFP. But also it is a nutrient base for the sperm, and that's why the sperm can actually survive, within the female tract up to 5 days. I mentioned this because she may have ovulated on day 1 and then thought that maybe by day 2, 3, 4, or 5, her egg has been discarded and she's no longer fertile. No, they could have had intercourse up to 5 days prior to ovulation, and she could still get pregnant because that sperm is just hanging around for 5 days on average. Not all, but some, the more vigorous ones. And so we have to leave a window of 5 days if that couple is seeking to avoid pregnancy, all right? Have to leave a window of 5 days before the ovulation. Okay. Let's move forward, because as you can see here, this zygote now will start splitting through mitosis. It's called cleavage, or the splitting of the cells, and it will cleave, it will split in half, right? From one mother cell to daughter cells. Each those 2 daughter cells will split again into 2 more daughter cells, and so forth. So we start getting into a geometric progression. 1, 2, 4, 8, 16, 32, 64, 128. I know it by heart already because I've said it so many times. So 1, 2, 4, 8, 16, 32, 64. Okay, in 7 splits, all right, in 7 generations, we already have 64 cells. Mm-hmm. Right. 16, 32, 64. That's called geometric geometric progression. If it were a linear progression or an arithmetic progression, what's an arithmetic progression? 1, 2, 3, 4, 5, 6, 7. So with an arithmetic progression, after 7 generations, we'd have only 7 cells. With a geometric progression, after 7 splits, we have 64 cells, and then it duplicates again and again, so you can see that the number rises exponentially very quickly. That is the 5,000 times that I'm talking about, how that embryo gets to grow so fast, so early, okay, because of this geometric progression. That happens through the voyage, if you will, the passage of the early embryo, all the way up to the endometrium, the inner lining of the uterus, until implantation, which on average is about 5 to 7 days after fertilization. Again, I make emphasis on this because I'm seeing ideology filtering into many sectors of society. There's certainly plenty of ideology in politics, for example. There's ideology in economy and many other fields. There's ideology in psychology and so forth. Now there's ideology in biology, okay? Yes, and I have now seen texts that are trying to maintain that fertilization is not complete until implantation happens, which is a big lie. It's just an absolute lie. It's unethical. It's dishonest because fertilization and implantation are 2 separate events. You can certainly have fertilization without implantation if the embryo is discarded, but you cannot have implantation without fertilization. And so to say that fertilization is not complete until implantation is just pure ideology to justify the manipulation of embryos that will never implant. For example, in an in vitro clinic, that the spare embryos will then be used for genetic manipulation and for embryo experimentation. And they'll say, well, it's not fully human because fertilization has not been completed because there's no implantation. And that is totally ideological. And I've actually talked to the 2 main in vitro clinics in town, one which is private and the other one is a Baptist hospital. And these are the geneticists, the scientists that are running these clinics say, no, it's just a little piece of tissue. There's no implantation. How can you say it's a human? So they're conditioning humanity to the place where the embryo is, to whether there's an implantation or not, okay? Which is totally subjective and arbitrary. Why? Why not cephalization, for example, when the embryo actually starts developing a brain? Or why not the heartbeat? Those are totally arbitrary assignments of when human life begins, but it doesn't match the biology. And these are the clinicians, okay, who studied this in luxury or detail much more than I know it because of their studies. Okay, so that's what we're up against. So you can imagine if those people are confused, what can we expect from the general population, right? Which actually think that these in vitro clinics are their saviors because they procure a baby for those who couldn't have the baby. All right, so here's the zygote. And you can see that syngamy has not been completed yet. You can see very clearly the 2 pronuclei there, all right? We don't know which is which, but there are the 2 pronuclei. In fact, you can also see the polar bodies here embedded into the zona, and then the corona out here. These are micrographs. In other words, these are electron photographs, not diagrams. These are actual photographs. So after the zygote splits, we start getting the 2-cell stage, the 4-cell stage, 8-cell stage, right? And the individual cells here are called blastomere. It's just a technicality, but all these are diploid. These are all diploid and they're all undergoing differentiation. They're all undergoing mitosis. This 2nd stage is called the morula. So now zygote was in Greek, now morula is in Latin. Morula in Latin means tiny berry. The original is mora, and just like in Spanish, una mora, okay, zarzamora, and in English, a berry like a raspberry that has its little clusters. So 150 to 200 years ago, when the early scientists were looking at these under the microscope back then, and they would see under the light microscope, which was the only thing available, they would see the little cluster of cells, the blastomeres, and would say, oh, morula, they look like little tiny berries, and the name just stuck. So it means a small berry from mora. So that's the 2nd stage, if you will, until we get to Oh, and here what happens is a process that is called compaction. You can see that it is timed. 48 hours, you're already into the 8 cell stage, right? So the first 24 is the first split, the first day, and then split again and split again. So by the second day, we're already into the third or fourth split of cells. But at the same time, because of the zona and the corona, the embryo itself maintains the same overall size, the same diameter, if you will, okay? Because it's still inside this soft shell. And so the individual cells, the individual blastomeres are each smaller and smaller. They're smaller, okay? So there's a reapportioning of cytoplasm, of the original cytoplasm, if you will, and that's just compaction. It's also compaction, but now this is cellular compaction in contrast to chromosomal or DNA compaction, which would happen when the chromatin of the chromosome condense in the supercoiling, okay? It's the same word referring to the same general process of tightening together, but applied to cells instead of to the DNA of the chromosomes. Anyway, just details. Eventually, we get to the third stage of embryonic development, which is the blastocyst. The blastocyst. Okay. What is interesting about the blastocyst is that now we have 2 cell layers. In other words, at some point late, after around the first week, more or less, between 5 to 7 days, the morula will start hollowing out on the inside. Will start hollowing out, okay? The morula. Now, that hollowing out, what is this? Is this air in there? Absolutely not, because there's no air. This is not exposed out to the atmosphere. There's no air to be sought. This is fluid. It's mostly water, okay? But it's fluid inside. But the point is that the morula starts hollowing out, and then we get 2 different types of cells. We get a cell layer that is on the outer layer of the blastocyst, okay? And that's called the trophoblast. And then we get an inner mass of cells, the inner mass of cells that is called the inner cell mass, or ICM. ICM. It's also known at times as the embryoblast. Here are different examples. Here's the embryoblast or the ICM, and here's the trophoblast, okay? This is the first level of cellular differentiation. First level of cellular differentiation. So that the trophoblast will have one function and the embryoblast will have another function. The embryo proper comes from the embryoblast, from the ICM. So the embryo proper, the next stage, the gastrula, will form from the embryoblast, okay, from the ICM. The trophoblast, in contrast, will become eventually the placenta, because the trophoblast is the one that is in direct contact with the endometrium. So we go to the next stage. Here is implantation. Here is the blastocyst. You can see that the embryoblast is inside, and the trophoblast is the layer of cells of the embryo that is in direct contact with the mother's tissue, with the endometrium of the uterus, okay? In the meantime, and so here you see the beginnings of a placenta, and also simultaneously there will be an increased vascularization on the mother's side, capillaries that are reaching out. Now this is a very interesting process, this implantation, and it's not altogether easy because the mother's tissue has a dilemma, it's in a quandary. This tissue is 50% compatible with the tissue of the mother, meaning the genes that came from the egg, but is 50% incompatible. The genes that came from the father are foreign, the same species, but genetically foreign at the level of detail, just like when we get an organ in an organ transplant, an organ from another human being is seen as a foreign organ by the recipient, by the patient, from the same species, okay? So it's at the level of genetic detail precisely in the variations, in the variants. That's where the issue is. And so the endometrium doesn't really know what to do with this item, with this blastocyst, if it's compatible or incompatible. And so the blastocyst has to convince the endometrium tissue that it is compatible, to not reject it, that it's not a foreign body, that is benevolent. Okay? Now, if we're going into an antagonistic model of going against each other, the mother could say, this is a parasite, and I don't want this parasite because I am entitled to my own blood and to my own nutrition. I don't want to give nutrition to this parasite. I get rid of the parasite. That's an antagonistic model. And in my opinion, that is where radical feminism is. There's absolutely no dialogue. I'm in absolute control of my body and I don't want this parasite. The law has allowed that to happen. Very sad, but very real. Because it's not a parasite at all. It is her child. It is her child. All right? It is her child. And so the decision to not have the child should have been prior. About 2 weeks prior, 1 or 2 weeks prior, at least 1 week prior. Okay? Anyway, so you see the vascularization that happens, and so this is all done in a very elegant biochemical way, that the embryo convinces the endometrium to allow him or her to embed and establish a placenta, a network of communication, which will become very organized, and capillaries on both sides, as the embryo continues to develop, and the embryo will then absorb nutrients from the mother and release toxins, at least originally, until the kidneys are somewhat more functional, but also the gas exchange, which in a sense is kind of more important or more immediate, meaning gas exchange, meaning that this embryo is going to need a lot of oxygen big time because of all the cellular growth that is happening. So a lot of oxygen to get those ATPs to do the work, right? And in exchange, the embryo will release CO2 that will be taken out into the bloodstream of the mother, and together with the maternal circulation will go out through the lungs, just like she processes her own CO2. All right? So there's more than an antagonism, there is a commensalism that happens here, but it's somewhat delayed in the sense that this is the way for a new adult. One way of looking at it is the way for an embryo to make another adult, all right? It needs that time, it needs that process to happen. So kind of an investment initially on the mother's side. Hopefully this kid will be the one who take care of his or her parents when they get old. If everything goes according to plan. All right, the fourth stage then is gastrulation. Gastrulation, which at that point, that inner cell mass starts differentiating further. Of course, the outer layer, the trophoblast, is not shown quite correctly here because the trophoblast has also developed into the syncytial trophoblast and they essentially established in the placenta. But the ICM has also undergone more cellular differentiation, essentially into 3 layers, okay? So the early process of gastrulation here is to establish 3 germ layers of the gastrula, the early gastrula, and those germ layers are just going to be ectoderm, mesoderm, and endoderm. Okay? Ectoderm, mesoderm, and endoderm, they're known as the germ layers, and it is from these 3 germ layers that all of the organs and tissues will form in the body. All of the organs and tissues of the body will come here from these 3 layers. We have essentially about 220 cell lines, they're called cell lines, the ones that make up organs and tissues, right, the cell lines. The dozen organ systems that we have, respiratory system, circulatory system, digestive system, muscular, skeletal, nervous system, etc. They come from these 200 lines, more or less 220 lines. Those 220 lines come from these germ layers. So for example, the easiest one, the skin, would you think it comes from the ectoderm, mesoderm, or endoderm? Ectoderm, right, the outside layer. So the outside layer will eventually become the skin. Whereas the The mesoderm, for example, will be mostly muscle and bone. And then the endoderm is mostly the internal organs, especially digestive organ and all the other internal organs that we have, especially in the trunk of the body. But an interesting detail is that the nervous system also comes from the ectoderm. And if you think about it anatomically, the nervous system is the innermost system of our body because it's encased not only by skin and muscle, but even by bone, all right? The head, the cranium, the brain, and then the spinal cord inside the vertebrae, which is bone, and it's encased in there. But how did the ectoderm get in there? Well, precisely by this process of pouring in of cells down the groove, the mid groove that goes lengthwise on the gastrol, as you saw on that movie, all right? And that pouring in, which is also called an invagination, all right, of the ectoderm as it pours in, and then the groove seals on the top. And when you end up is a neural tube inside, which is then gets encased by bone, and that will form the nervous system. The head end will cephalize, will become larger and become the parts of the brain, and then the rest will become the spinal cord, and the vertebrae will segment so that it's articulated, and it offers rigidity and articulation at the same time. So it's a very, again, elegant design, for lack of another word. All this programmatic, it happens every time over the decades and centuries and millennia that we have been around, from generation after generation, we have having us to tell it to do anything. It just happens because it's programmatic that way, or because of the genes that are in the DNA that program all this development. And so those are called developmental genes. They express at different times of the embryonic development and are in charge for all the segmentation and the differentiation that is happening along the way. Okay, it's just a diagram of a cross-section of the blastocyst implanting and how the early placenta begins to establish, making contact with capillaries in the vascularization of the mother. In the meantime, the embryo continues to develop through gastrulation into the further process. Here is the neural groove that I was talking about. And this is a longitudinal, now photographed. These are micrographs, electron photographs. You can see the 3 germ layers here, endoderm, mesoderm, the neuronal tube as it's sealing on the top, okay, and the mesoderm in the middle. Again, a little too much detail, but all I'm trying to point out by this is that there's a gradual process that is happening. It continues along the way, organically and gradually for, like I said, the first 8 weeks. This, by the way, are the Carnegie stages of embryonic development, which is the standard in medicine today. These models, again, as you can see, they're photographs of the embryonic development. By the time the embryo is 8 weeks, all of the main organ systems are in place, but in miniature, because the embryo from head to rump, as they say, is about 1 inch long, okay? And at that point then, the embryo changes name, technically, to fetus after the first 2 months. And for the next 7 months, it's mostly growth in size. There's more development, of course, and specialization, cellular differentiation at the more detailed level. But as far as the organ systems, they're in place, okay? And it's mostly cellular growth then for the next 7 months. And the change in name is symbolic also, representative, because fetus, back to Latin, means a tiny being, tiny being. Okay. Questions, comments?
I have a question. Yeah. How does the RH immunization happen and at what stage?
Okay, so RH is one of the factors that is in the blood, specifically in the RBC. Let's take a moment to look at that. It's called RH factor. And this is how it works. So it's a capital, so maybe just maybe this one. Well, this one actually has the the blood groups and reasons. Okay, this will work. So this is what we have. There, on the surface of the RBCs, the red blood cells, which are in the bloodstream, right? Okay. The surface of the red blood cells has a number of proteins, but for the purpose of the blood type and the RH factor, there are 3 proteins of interest, okay? 2 proteins are paired and 1 protein is by itself. That other protein, we either have it or don't have it. Now, the protein that we either have or not have is the RH protein, okay? So all our RBCs, we either have that protein or don't have that protein. So if we have the protein, the RH protein, does that make us RH positive or negative? Positive, because we have the protein. Meaning that the people who don't have that RH protein on the surface of the RBCs, then they are RH negative. Simple as that. Where does RH come from? RH comes from a monkey, which is known as the rhesus monkey. It's the genus name of the monkey. All right. And why the rhesus monkey? Because this is an old world monkey, middle-sized, that was used earlier in science, in medicine, for exploration. And before they went to clinical trials on humans, they would experiment on the monkey. In case the drug didn't work, the monkey would die and not the human would die. Okay, so before getting to the clinical trials, they used mammalian trials. And this monkey, I guess, I don't know, it's fairly benevolent, easier to keep in cages and whatnot. And it was just chosen for any number of characteristics, which I'm not familiar with. But this monkey happens to have that protein on the RBCs, okay? So they just use the first 2 letters of the genus. This is rhesus monkey, and the first 2 letters are RH, and that's where the name comes from. So, and it's essentially the same protein that we have, very similar. So if we do have the protein, we're RH positive. If we don't have it, we're RH negative. It may be, I personally don't know of a function for that protein. They may have it, but it may be a vestigial protein also pointing to our mammalian. This is what we will call compatibility with other mammals, especially at the level of primates, right, with monkeys. And so this is another evidence for evolution, the fact that we have that protein also. Anyway, back to the factor then. So you know how it is established, and it's just an inheritance factor. So if either our father was RH positive or our mother was RH positive, then there is a 50% chance that we would inherit that positivity, unless there was some recombination occurring at that site that codes for that protein. Okay, so that's how we become RH positive or RH negative. Now, for RH positive people, you know, we can receive, let's say, I'm RH negative, but for an RH positive person, can that person receive RH positive blood? Yeah, because they have the protein, and if they receive blood that has the protein, it's no problem because their body is used to that protein. What about RH positive person who receives RH negative blood? Well, that blood donation, that transfusion, doesn't have the protein, but who cares? The patient has the protein, and if he receives blood that doesn't have the protein, no problem, okay? But the converse, the RH negative people, all right, we can receive blood from RH negative because no protein in that transfusion. But if we receive blood from an RH positive transfusion, what happens? I don't have the RH protein, and so my body is going to recognize that protein as a foreign body, and it's going to attack it. It's going to mount an immune rejection on every single RBC that I get on that transfusion. And so I'm going to start forming clots of my antibodies trying to shut down that foreign body, which is the RH factor on the RBCs that I'm receiving as a transfusion. So I'm going to develop a reaction, okay? That can happen with babies if the mother is Rh negative and the baby is coming Rh positive, she's going to start mounting an immune reaction, all right? Now, as far as I understand, she can tolerate the first pregnancy, right? But I think by the time the second pregnancy comes along and it's the same scenario, then that second baby is in danger because the immune reaction is going to be too strong and can interfere with the pregnancy, or the baby will become deficient in blood, and they're called typically blue babies. In other words, they're born with a deficient amount of RBCs, and an intervention has to be done. So that's the issue with the Rh factor, all right? It depends on who's positive and who's negative of the parents and what type, what factor the babies carry. In addition to that, then there is also the blood type or the blood group, which are 4 groups, and this is in addition to the Rh factor. Okay, so we can be positive or negative with regards to the Rh factor, and then there are 2 other proteins that are mentioned that are just called protein A and protein B, or antigen A and antigen B, and they are also on the surface of the cell of the RBC. So if we have protein A, then we are A-type blood. If we have protein B, we are B-type blood. If we have both proteins on the surface of the RBC, then we are AB blood. And if we have neither of the two, sorry, okay, no, stop. If we don't have either A blood or A protein or B protein, then we are 0 protein, which we call O, but the O group is the 0 group. In other words, we don't have either A or B, aside from either having or not having the Rh factor. See how it works? So, bottom line, we can be A positive or A negative, B positive or B negative, AB positive or AB negative, and we can be O positive and O negative. And then the same principle applies. For A blood people, they can receive a transfusion from A, and they can receive a transfusion from O, but they cannot receive a transfusion from B because this B protein is going to look foreign to the A individual. And they cannot receive transfusion from AB either because the B part is going to look foreign. The same argument can be made for B. Then for AB, AB can receive blood from whom? From AB, from A, from B, and from O. So AB is a universal recipient. But O is stuck because O can receive from A? No. Cannot receive from B? Cannot receive from AB? Can only receive from O. So O is called universal donor. But at the time of receiving blood, they're stuck because we can only receive from O. It happens, and thanks be to God, the vast majority of blood type is O type. So it's the most common one. So it compensates because there are more individuals with O type than most individuals are universal donors. And that's a good thing when the universal donor needs a transfusion because generally there are more O type blood in any given population of humans, okay? And the different ratios, I forget which is the most rare of them. I think it's AB, but I'm not sure, okay? But there are different ratios in the population. And that is balanced out again through evolution has been balanced out so that the ones who are more difficult in receiving blood are the most by the number of individuals. So 2 different issues. One is the blood type, A, B, AB, or O, and the other one is the Rh factor, either positive or negative. They can all affect pregnancy one way or another. Okay. So here's the whole development. And again, the whole point is that because this is a continuous organic development of organs and tissues and differentiation and growth in size and shape and so forth, there's no substantially distinct moment when this individual passes from non-human to human, okay? He or she is carrying the same 23 pairs of chromosomes as he or she started with at fertilization, all the way to the trillions of cells that he or she has accumulated within 9 months by the time of birth. Okay, so now I have another little movie here. Let me see any questions or comments. Anyone from the online? All right. Now, this Tetrads, Alexander Tetrads, this fellow was a computer scientist, a mathematician. He developed the algorithms for this little movie, all right? And the other fellow that did the photographing and so forth, he'll explain it, went on to win a Nobel Prize. But it was a teamwork, and Tetrads is the one who actually did the algorithm for visualizing this, which are going to be photography. It's actually not photography proper, they're sonograms that are colorized, all right? It's a sonogram, which means that these are real live takes on live individuals. And I'll wait for Dr. Penelva to come back and just wrap it up. Yes, it's one of these TED Talks, right? It's only about 10 minutes, but I thought it was fascinating how this fellow is giving this talk and showing his slides. Again, he did the computer algorithms to be able to visualize and colorize these sonograms or ultrasounds that we're taking on pregnant moms. And what you see is the gradual development of the different organ systems, because he gave a different color or tone to the different organ systems. And you'll see that how the heart is developing, the liver, the different organs, the brain, and so forth. It's just fascinating. And to me, what is of value is that these are actual sonograms. You know, they're not drawings or caricatures or anything like that. This is how it's happening live in nature. Okay, so let's just give Dr. Penelva a moment. Like I said, it goes on for about 10 minutes. He's a little bit of a fast talker, but you can follow along. Yeah, for this one, I need the sound on because he's actually explaining. While we wait for Dr. Peña, can you, sorry, can you hear okay? Those of you who are connecting through the Zoom? Let's see, turn this on, yeah. Okay, you can hear okay, yes, good, all right. I hope you can hear the video also. It should go through the same system. Okay, thanks. Okay, well, she's there at the door. Let's see, let's go forward.
I was offered a position as Associate Professor of Medicine and Chief of Scientific Visualization at Yale University in the Department of Medicine. And my job was to write many of the algorithms in code for NASA to do virtual surgery in preparation for the astronauts going into deep space flight so they could be cut in robotic pods. One of the fascinating things about what we were actually working on is that we were seeing, using new kind of scanning technologies, things that had just had never been seen before. I mean, not only in disease management, but also things that allowed us to see things about the body that just made you marvel. I remember one of the first times we were looking at collagen And your entire body, everything, your hair, skin, bone, nails, everything is made of collagen. And it's a kind of a rope-like structure that twirls and swirls like this. And the only place that collagen changes its structure is in the corner of your eye. In your eye, it becomes a grid formation, and therefore it becomes transparent as opposed to opaque. So perfectly organized a structure, it was hard not to attribute divinity to it, because we kept on seeing this over and over and over again in different parts of the body. One of the opportunities I had was one person was working on a really interesting kind of micromagnetic resonance imaging machine with the NIH. And what we were going to do is scan a new project on the development of the fetus from conception to birth using these kinds of new technologies. So I wrote the algorithms in code, and he built the hardware. Paul Lauterborough then went on to win the Nobel Prize for inventing the MRI. I get the data. And I'm going to show you a sample of that piece from conception to birth.
MRI is the magnetic resonance imaging, okay? And so it's using magnetism to visualize internal organs, which is essentially minimally invasive, because even there's electromagnetic waves going through the room right now. And so they're innocuous to the body, and it's a way of visualizing the internal organs. But of course, none of that can actually be seen unless we have the algorithm code, the actual computer code, to decipher it and to visualize it, to make it into an image that we can actually see. Otherwise, it just remains an electromagnetic wave. And that's what this fellow Sarias did, all right? So here we go. Here's the egg. And the complement. Fertilization. First mitotic division. Second. Third, etc. The embryo blastocyst. You have the early gastrula. It's actually moving, the heart is beating early on already. You see the limb buds, the somites of the spinal cord forming, circulation. See the tail bud, which is then reabsorbed. You see the eye, the ear, the mouth. Again, looking internally at circulation. See the beating heart. External surface of the embryo, fetus now. Different parts of the brain as they're forming. This is time-lapse photography. Not photography, sonography. It's close to 9 months. Birthing, this is actual birthing. He only has the pelvis of the mother, the skeletal structure. Look at that, this is live. The rest of her body is transparent to the algorithm. Now the baby is going to push. They push against the acetabulum of the pelvis. There. Push up. Literally. They push. See. They push against the. See. They push against the coccyx of the mother. And there's baby inside and out. Look at that. Halfway. Amazing. Just pause for a moment. New York State just legalized killing that child.
New York.
Yeah, New York. The state of New York just legalized abortion up until the 9th month. Until the baby, 9 months. Unless the baby actually sticks out his head or her head outside the woman, that baby can be killed legally in New York State. It just passed.
Thank you. But as you can see, when you actually start working on this data, it's pretty spectacular. And as we kept on scanning more and more, working on this project, looking at these two simple cells that had this kind of unbelievable machinery that will become the magic of you. And as we kept on working on this data, looking at small clusters of the body, these little, pieces of tissue that were the trophoblasts coming off of the blastocyst, all of a sudden burrowing itself into the side of the uterus, saying, I'm here to stay. All of a sudden having a conversation of communications, the estrogens, the progesterones, saying, I'm here to stay, plant me, building this incredible trilinear fetus that becomes within 44 days something that you can recognize, and then at 9 weeks is really like a kind of a little human being. The marvel of this information, how do we actually have this biological mechanism inside our body to actually see this information? I'm going to show you something pretty unique. Here's a human heart at 25. It's just basically 2 strands. Like this magnificent origami, cells are developing at 1 million cells per second at 4 weeks as it's just folding on itself. Within 5 weeks, you can start to see the early atrium and early ventricles. 6 weeks, these folds are now beginning with a papilla on the inside of the heart, actually being able to pull down each one of those valves in your heart until you get actually a mature heart, and then basically the development of the entire human body. The magic of the mechanisms inside each genetic structure saying exactly where that nerve cell should go. The complexity of these, the mathematical models on how these things are indeed done are beyond human comprehension. Even though I'm a mathematician, I look at this with the marvel of how do these instruction sets not make these mistakes as they build what is us. It's a mystery, it's magic, it's divinity. I mean, then when you start to take a look at an adult life, take a look at this little tuft of capillaries. It's just a tiny substructure, microscopic, but basically by the time you're 9 months and you've given birth, you have almost 60,000 miles of vessels inside your body. I mean, and only 1 mile is visible, 59,999 miles that are basically bringing nutrients and taking waste away. The complexity of building that within a single system is again beyond any comprehension of any existing mathematics today. And then instruction set from the brain to every other part of the body. Look at the complexity of the folding. Where is this intelligence of knowing that a fold can actually hold more information? So as you actually watch the baby's brain grow, and this is one of the things that we're doing right now. We're actually doing a longitudinal studies of actually scanning babies' brains from the moment they're born every 6 months until they're 6 years old. We're going to be doing actually about 250 children, watching exactly how the jidai and the sukai of the brains fold to see how this magnificent develop actually turns into memories and the marvel that is us. And it's not just our own existence, but how does the woman's body understand to have genetic structure that not only builds her own, but then has the understanding that allows her to become a walking immunological cardiovascular, system that basically is a mobile system that can actually nurture, treat this child with a kind of marvel that, is beyond, again, our comprehension, the magic that is existence, that is us. Thank you.
Tremendous, huh?
Yes.
Yeah.
I have a question.
Yeah.
What about the soul? Do we consider the soul
Very good.
since fertilization or?
Exactly. So that is what I meant by the ontological status of the human embryo, okay? What about the soul? Because again, even though he's using magic and mystery in his dialogue, it's analogical in the sense that as complex as this development is, it is an organic complex and it has an explanation, ultimately it has a biochemical explanation. Everything has to happen in place for the next stage to go on, right? But the soul is different because the soul, the one thing that the soul is not, for example, is not complex because it's not made of parts, it's simple, all right? And simple means that it's also, that it can perdure through time. And so it is our belief, now we go into a system of belief, but it's our belief that the soul has an origin, but has no end in the sense that it is eternal. Mm-hmm. And so that requires the divinity to intervene directly and individually at the level of creating a soul for each one of us. So we can ask the question, and I'll leave it at this because that's what I'm going to pick up next Saturday, okay? When does God intervene directly? In other words, where is the matter human for that matter to receive the form, the information of the soul, of the human soul, so that we have matter and soul one, all right?
Because last semester we also spoke about the burial.
Yes.
Now in the ancestry, so basically,
exactly.
You start to bury a lady when it's after it's born.
Yes.
If you lose it spontaneously in a fetus, you don't.
We don't have to bury, but now some couples may want to bury their miscarried child, all right? If there is some organs and visibly formed already, even though that embryo or fetus is small, they can be buried. There's no particular set time, and we've gone back and forth on this with Respect Life, you know, and we cannot become over scrupulous either, because medically it's a dead fetus or dead embryo is technically medical waste and will be disposed clinically in that way, which typically medical waste is incinerated just for health reasons, okay? Mm-hmm. But if couples want to bury their unborn child, there has to be a cutoff point, you know, less than maybe 3 or 4 months. It's just too tiny to bury, right? But beyond that, once that little fetus has a size, a visible size, they can be buried. Mm-hmm. And it's up to the couple to do that. They'll have to make arrangements and so forth. But yes, yes. But if the baby has died, then that soul is no longer there. Okay, just like with a human being. Now we're talking about a cadaver. So we have the organic remains of a human being, but the soul is no longer there. The soul is in the mystery of God, in the presence of God. You know, He decides. Emma.
Do you think that they have their own personalities? Because when I used to scan twins, one of them was more calm than the other, and the other one was more hyper. Yeah. So it was kind of hard for me to get good pictures of hyper one.
Interesting, right? Stay still. Come on, kid, stop it. Look at the camera for a moment. Yes, sure. So yes, absolutely. They do have an individual personhood, all right? And I'll talk about that at the beginning of next class for sure. But basically, you can think of this. So the different ways of looking at personhood, you're looking at it a little bit from the psychological perspective of behavior, of movement. But we can also look at personhood from the philosophical point of view, and that's the ontological. When can we attribute personhood to the human person, right? Before birth or after birth, when they get their driver's license, or when they marry, I mean, when do we become persons? Which again is a difference anthropologically speaking or legally speaking, because we know that legally the law defines legal personhood once we're born, but not before birth. And that's precisely the problem. The big problem with abortion is that the law, the civil law, okay, what we call positive law made by human beings, simply does not recognize the unborn human as a legal human, all right? It's not the only case. For example, an illegal immigrant is not a legal person in the United States. It's an illegal person, okay? So they don't have a legal status, kind of analogous to the unborn, but they're certainly a human being. And that's the contradiction, because they too are protected by law. It's not that we can kill illegal immigrants because they're illegal, all right? But they don't have a legal status. So there are so many contradictions built into the legalization of abortion that is just sheer ideology of a group that wanted to impose their way and essentially bamboozled the rest of the population, or a large segment of the population, to go along with it. Mm-hmm. And these are the challenges that we have, trying to convince these people that, with all due respect, they were wrong about the humanity of this unborn. Mm-hmm. And that's the big challenge. That's the big challenge. So we'll get into installment, what is known as installment, either direct installment or immediate or immediate installment, all right? Direct or delayed. So this is just basically kind of a summary regarding embryonic development. There's a continuous development of tissues, organs, and systems, all the way from the zygote, from fertilization, up until birth. Even after birth, development doesn't stop there. Okay? The child is only a foot long, has to get to about 6 times larger, has to reach biological maturity and all that, which is defined in terms of sexual maturity, biologically speaking. But we see that there is continuous growth and development. So if we're not, see, I'm just trying to set the stage as logically as possible, that if we don't grant the fetus, the human fetus, humanity because it is not fully developed, well, the baby is not fully developed either. You know, you put out any newborn, leave them out on the sidewalk there, it's not going to last long, you know, before that baby is going to die. So it's still dependent, very much dependent, totally incapable of feeding herself or himself. And so it's not a question of dependency, it's not a question of development, because we are born underdeveloped, we still need to mature, and so forth. So these are the internal contradictions that point to the fact that it's an ideology that has been forced on society, and mostly out of ignorance on the other part of society, the biological ignorance, which should have been learned in high school, right, in sophomore biology. Okay, so, well, it's 12:20. We can stop here and pick up next Saturday, all right? And I'll pick up with this ontological status, the ontological status, which will get a little bit into philosophy and anthropology, meaning when do we start becoming human? When do we become a human being as such, all right? And I have some slides also to illustrate that, human individual, human being, human person, and all this. Well, thanks again, and we'll end here. And God willing, see you next Saturday. Be attentive to an email with regards to the entrance, 37th or 32nd, right? And it may probably depend on the weather, whether they'll close it or not. Okay, I'm going to stop recording now. So, Giselle, Monica, thanks again. And Father Jose, also, I'll be YouTubing this as usual and sending the link a little later today, okay? Thanks again.
Thank you,
Bye.
Father.
Okay, thanks a lot. Bye-bye.